Hello everyone, apologies for today. I've been having some issues with my screen recording software and some updates and everything like that. So I'm still working on getting my face cam working again. Just a couple issues with that. I should have it resolved by chapter 3. But I wanted to make sure that this video got posted so that everyone could get ready for the quiz and fill in everything regarding the discussion. And then move on to chapter 3. So as we talked about really briefly in chapter 1, I talked some about the biological underpinnings of cognition. And what chapter 2 focuses on is the research that's being done regarding these biological components to cognition. So this area of study is called cognitive neuroscience. Now, some questions to think about as you are reading through the chapter, as you're completing the quiz and going over your chapter notes. One of the things to think about is what is cognitive neuroscience and why is it necessary? So we'll talk a little bit about why understanding the biology of the brain is important regarding cognition. Also, how is information transmitted? Cognitive neuroscience focuses on a lot of the actual mechanisms that allow our brains to function and how information is transmitted through neurons and through parts of the brain. Also, how things in the environment, like any sort of visual representation that you have in your brain, how is that actually represented, and how can we attach different theories regarding representation to different parts of the brain? And just in general, what does studying the brain tell us about cognition? Knowing what different parts of the brain do, knowing how they function, what does this tell us regarding all of those different components from perception to attention to memory? Now, basic definition time. Cognitive neuroscience is the study of the physiological basis of cognition. And this focuses not only on the individual components of the nervous system, but also the entire system and how all of these little pieces interact with each other. And it's important to consider that we use cognitive neuroscience in collaboratively with other areas of cognition. And this is important in every science, but especially in cognition, because when we use multiple perspectives to study our behavior and how our brains work, we can take all of these different viewpoints and put them all together and try to figure out in a bit more detail how cognition works. These two examples that you'll also see in your book rely on letter A references all the chemical processes that go through our brains in order to create our perception of objects, and then we can attach that to letter B, which is theories regarding memory. And so being able to attach these biological processes to behavior is one of the main focuses of cognitive neuroscience, and one reason why it's not just neuroscience. While neuroscience is very important, a cognitive neuroscientist is always going to focus on how these the biological basis of cognition attaches to the behavioral basis. Now, if you've taken any sort of biological psychology course or any biology course that focuses on the brain, we usually start with the nervous system and neurons. Now, the importance of neurons is that these are very specialized cells, and they have a lot of little parts that all interact within the neuron and between neurons to actually generate thought processes. Now, all of these individual neurons are important, but the most important function that they undertake is this idea of a nerve net, and the idea that all of these neurons working in tandem basically allow us to think. And this constant communication between neurons is something like a highway in which every street connects to one another, but there are no stop signs. So you have the almost instantaneous transmission of information on a nonstop basis. Now, one important thing to note in terms of the development of cognitive neuroscience and neuroscience in general is that there are always alternative perspectives. And one of those important perspectives that's talked about in your book is this perspective espoused by Ramon E. Cajal. And this is called the neuron doctrine. And this focuses on the idea that individual nerve cells transmit signals, and they're not necessarily continuous with other cells. Now, while it's important to consider the activity of individual neurons, and we'll talk a little bit more as this lecture goes on about that, it's also important to acknowledge that it doesn't necessarily undermine the idea of a nerve net, but it does refocus our attention on the function of individual neurons. And here's an example of what a nerve net looks like under an electron microscope. And the one on the left is focused more on the actual net-like web structure that the nerves take up. And letter B, that arrow is actually pointing to the cell body of a neuron. And the third example that I have on this page is in reference to computer science methods that are used to kind of simulate how our neural net works. And this is something called a Markov model, in which we have inputs, and you can think of each gray circle as a neuron, and all these neurons interact with each other and then prompt an output. And so we can kind of think of that in terms of inputs being sensations, our input layer being our things from our eyes to our ears and like our optic nerve and those parts of the brain that process that information. And that hidden layer is the area that we're really trying to establish where activity is going on in the brain. And then we have the output layer, which can be likened to the idea of muscle output or movement of the eye or any sort of behavior that we might enact as a function of the sensations that we feel. Now, the parts of the neuron, or the three of the most important parts, are the cell body, and basically these keep the cell alive. The dendrites, these are the parts of the cell, the kind of branch-like structures on the cell body that receive information. And then the axon, which is kind of the cabling for each neuron that actually transmits electricity down the neuron to the axon terminals. Looking back at that diagram that I posted earlier, those three most important areas being the cell body, the dendrites, and the axon, moving all the way down to the axon terminal. And I have a nice video that's going to describe a bit more about how the neurons function. Now, to preface the video that we're going to watch really briefly, the way that neurons communicate is through this thing called action potential. Now, in this case, we have an activation of the neuron as it receives signals from the environment. That information that in the form of electricity travels down the axon to basically end at the axon terminal, and then that spreads out to other neurons. And one important thing that a lot of cognitive neuroscientists and neuroscientists do is they measure these action potentials. And an important thing to note is that we can actually measure the activity of individual neurons by attaching microelectrodes that can pick up electrical signals very near to the neuron. And we can see that these neurons can activate extremely rapidly. They're active for approximately a second, but it's a very rapid flash of electrical output. And so I'm going to add a little video. I'm going to turn off the text at the top of the screen, and we're going to watch this real quick. This is from the series Crash Course that's done by John Green, sorry. No, it's Hank. Apologies. Hank Green is a Crash Course series, and there's an entire series on the nervous system, but this one does a really good job of demonstrating how action potentials work inside the cell.
What if everything you did, thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got Electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around -70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. neurons. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around -55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter like serotonin or a hormone latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at -70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about -55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about +40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to -75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around -55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization, and a brief bout of hyperpolarization. Thanks for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale, the script was edited by Blake T. Pestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe. One more thing before you leave. We like Crash Course a lot, and we hope that you like Crash Course a lot, but I kind of feel like Crash Course is only useful for a certain segment of the population. Like, once you get to a certain age, then it's good, and then forever, it can be helpful to people. But younger people, not so much. And so, we are creating Crash Course Kids, hosted by Sabrina Cruz from Nerdy and Quirky. Crash Course Kids will start out focusing on 5th grade science, but we'll keep expanding to other topics as the channel grows. Sabrina will be talking about food chains and gravity and how the sun works and how plants eat and why flamingos are pink and many other topics. Oh, and another note, teachers, you can rest assured that we've got you covered. There will be info about the standards we've used to make sure that we're doing our very best to help you out. So if you are a teacher, or you know a teacher, or you know a child, or you know someone who has a child, or you've ever seen a child, you can tell them to go to youtube.com/crashcoursekids and subscribe, and you can go do that as well if you would find that kind of content useful or interesting.
Okay, and just for reference, just as a reminder, that was Crash Course. It's done by the Green Brothers. Really spectacular channel. They do a lot of educational materials, as you heard him say at the end of the video. But focusing on the actual content of that video, it's really important to remember that this is an effort to establish a biological basis for a lot of the cognitive phenomena that we study as cognitive scientists. And so being able to attach this idea of intensity is very important, because there's a physical analog for the sensations and thoughts that we are trying to describe in cognitive psychology. And so just reiterating that idea that being able to measure an action potential relies on this idea that the amount of action potential isn't what matters. What matters is the rate at which this action potential is being achieved. And for low intensities, a slower firing of the neuron occurs, and for higher intensities, a faster firing occurs. And some examples of this low versus high intensity is shown in your book, but also shown here. The idea of light pressure, you can basically think of each one of these spikes as an action potential. And so, as you can see, the amount of spikes that are happening at light pressure are much less than those number of spikes happening at hard pressure. And this is a good way to be for our brains to be able to distinguish between different types of stimuli to establish whether or not we're holding a cup too tightly, or if something is threatening us, we're going to have a faster activation of that action potential and information being sent throughout our neural net. Now, the areas between neurons are called synapses. And even though we like to think about a neural net as this kind of interconnected bundle of fibers, it's important to acknowledge the fact that there is still a tiny gap in which neurotransmitters are released from one neuron that then move to the next neuron to activate it at the site of its dendrites. Now, neurotransmitters are the chemicals that affect this electrical signal, and they cross the synapse and then they bind with dendrites. And these come from the actual axon terminals that were shown in the neuron earlier. Now, moving from this idea of the individual neurons, Hubel and Wiesel are two scientists who wanted to study whether or not there's some sort of specificity that occurs at the neural level. And specifically, they wanted to look at feature detectors. And basically, they were trying to establish the biological basis of neurons activating for specific features in an environment. Seeing a circle, versus seeing a square. Their question was, do these activate different neurons, and which neurons do they activate? Now, here's a quick little video showing an example of some of the studies they did to basically track these feature detectors.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen and nothing seemed to work. And rather by accident, one day we were shining small spots, either white spots or black spots, onto the screen, and we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina, and every time we did that, we'd get a response.
Now, in looking or in watching that video, you heard these small pops that were happening when they were moving the transparency along the projector. Now, those pops are actual activations occurring within neurons that they had basically attached electrodes to in the cat's brain. And so this was one of the first most prominent studies that was done to actually establish where these basically where these feature detectors might exist in the brain and how we can actually track them. Now, these really simple feature detector experiments lead us up to this idea of processing information because, of course, as we all know, we all process more information than just lines moving across our field of vision. So an important area in cognitive neuroscience is looking at this idea of hierarchical processing and the idea that when we perceive different objects, we do it in a very specific order and it moves from lower to higher areas of the brain. Basically, we're processing information on the simpler end, and so very basic features of things that we might be seeing, hearing, receiving as sensations, and those move up to higher levels of complexity, more detail, when we're actually processing this information in our brain. And ways that we actually represent this information, there are three specific ways that the book talks about, and these are through specificity coding, population coding, and sparse coding. Now, a good example of each one of these types of coding is thinking about facial recognition. Now, how we recognize faces, we can use an example in terms of specificity to think about individual features of a face and the actual activation of a single neuron that differentiates between different stimuli. So in this example, we have Bill, Mary, and Raphael. We see their faces, and a different neuron activates for each one. Now, this is very specific and is typically associated with simpler stimuli, but there could be distinguishing features between Bill, Mary, and Raphael that will activate different neurons specifically. Now, this example of population coding relies a bit more on a multitude of neurons activating at different rates. So you think of the idea of that popping noise happening at a slower rate for some neurons and a faster rate for others, and then not happening at all for other neurons. And we can look at basically these activation curves, and they're all different for different stimuli. And then we have this idea of sparse coding that is basically relies on a fewer number of neurons than population coding, but more neurons than specificity coding. To attach it back to that video we just watched, specificity coding is typically used to describe very simple features, sparse coding slightly more complex, and then population coding more complex features in the environment. Now, moving from this idea of the neural net activating, we start to look at bigger parts of the brain, and this is called localization of function. And what that basically means is that cognitive neuroscientists look at different areas of the brain and what they're responsible for. Now, we can look at this by looking at how cognitive functioning breaks down when parts of the brain are damaged. And we focus primarily, in terms of talking about localization, on the cerebral cortex. So this is the area around the brain, and that's where a lot of the activity occurs when we're looking at different parts of the brain that are being activated. One of the most prominent areas in cognitive neuroscience where we talk about localization of function is in regard to language. Now, two of the areas that you might have heard about in general psychology or another psychology course are Broca's area and Wernicke's area. Now, Broca's area is located in the frontal lobe, whereas Wernicke's area is located in the temporal lobe. And one way that scientists studied these areas of the brain and basically came up with the idea that, well, there's an area that's specific to language production, and there's an area that's specific to language comprehension, is looking at people who had who enacted very similar behaviors, and then looking at their brains and seeing that they had lesions or parts of their brains that were damaged that all matched across subjects. And so, looking at Wernicke's area, people who had problem with language comprehension typically had damage to Wernicke's area of the brain. The same for people who had language production problems for Broca's area. Now, this is an example of someone with Broca's aphasia, and we'll watch a very small amount of this. But as you see from the very beginning of the video, the issue that she's having with communicating is very much a problem of producing speech.
So what's your name?
Scott. Oh, no. Sarah Scott.
That's right. And how old are you?
I can't.
Try.
I can't.
You're 19. 19. Yeah. And what happened?
So one of the big takeaways, even though this is a very brief part of the interview, is that the reason why Sarah has this issue with language production is due to damage that happened due to an ischemic stroke. And being able to understand why she's having these difficulties in producing speech is very important, not only in just understanding how the brain works, but also in helping to treat people who might have these consistent behavioral issues that are manifesting because of damage to their brain. And here's another example of a gentleman with Wernicke's aphasia, which is also sometimes referred to as fluent aphasia. And we'll see that that distinction between production and comprehension is very different, even though they're both issues regarding language.
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people for them over there. They're diving for them at the moment. They'll save in the moment. He'll have water very soon for him. With luck, for him.
So we're on a cruise and we're about to
We
get to
will
Juneau.
store it right here, and they'll save their hands right there for them.
And what were
So looking at Byron's problems with comprehension, he's producing speech that has what's called normal prosody, which is kind of the pace of speech, but a lot of it is nonsensical, and there are a few important terms that are kind of mixed in there, but it's difficult to try to dissect what he's trying to say, because he's doing something that's basically called word salad. And this is something that occurs across a lot of different disorders, not just with people who have damage to Wernicke's area of the brain. It's also a very common symptom of schizophrenia. And thinking about these two really salient examples of language production gives us a lot of evidence, basically to think about how all of the different parts of our brain are doing all of these different things. And when we damage them or we treat them poorly by doing different things that might be unhealthy and things like that, that can actually influence our mental state, or in the least bad situation, basically can mess up our behavior without basically move it outside of our control. Now, the four major parts of the brain that we talk about are the frontal lobe in the front, and then the temporal lobe, the parietal, and the occipital lobe. And we can distinguish these four lobes based on what they're responsible for in terms of what we've isolated due to looking a lot of the time at people who have had disorders related to brain damage. Now, the three parts in the back part of the brain, the occipital, parietal, and temporal, are primarily responsible for receiving information regarding the senses. So the occipital lobe is responsible for vision, the parietal lobe is responsible for touch and temperature and pain, this idea of pressure and sensation, and then the temporal lobe is primarily associated with hearing, taste, and smell. Now, all of these lobes work in tandem to basically allow us to receive sensory information and then process it. And the frontal lobe is responsible for coordinating a lot of this information. And a really interesting fact about the frontal lobe is that oftentimes in people who have disordered thinking or lack some level of behavioral coordination, oftentimes due to brain scans, we can establish that they have some sort of damage or deficit in their frontal lobe. So the importance of that front part of your head is very interesting, and we can see that that deficit occur in people who have had damage to their frontal lobe. Another thing, so basically on top of just these individual areas of the brain being influenced by damage, we can also look at how we have this double dissociation, which essentially means that sometimes damage in one area of the brain can result in one function being absent, while another function is unimpaired. And then damage to another part of the brain can cause function B to be absent, while function A works perfectly fine. And through this kind of inference and comparison across patients who've suffered damage to their brain, we're able to establish with a bit more resolution what parts of the brain are responsible for very, very specific cognitions and behaviors. Now, in thinking about how we organize this information, one of the most prevalent tools used to study brain functioning is called magnetic resonance imaging, or MRI. And what an MRI does is essentially, because all of our cells are very full of water, and especially because water is made up of H2O, we have hydrogen atoms inside all the tissues in our brain and our body. because this is not just something that's limited to the brain. But what we can do, because those water molecules exist in our head and in our body, we can actually, this giant donut-looking part right here, it's a massive magnet. And what that magnet does is it basically spins around and it creates a magnetic field, which we then use to resonate those protons within those hydrogen atoms. And the oscillation rate at which those hydrogen atoms fluctuate allows us to basically determine what the individual cells are doing. So excited hydrogen atoms emit a radio frequency, and we measure that using a receiving area in the MRI that's around that magnet. And those are switched on and off very rapidly, and that's the repetitive noise you might hear if you go in to get an MRI. And this contrast, so that signal that we're picking up, allows us to establish what different tissues are doing. And this allows us to see if there's tissue damage in parts of the brain, whereas the only other way we'd be able to establish that is if we actually did some sort of invasive surgery. This is just a GIF of a MRI scan, basically being able to determine the differences in tissue and how they act in regard to that magnetic field. And what the MRI does is it takes slices, and then we can use software to basically make a three-dimensional model of a person's brain and whether or not they have damage to specific parts. Now, trying to put all of these little areas of the brain together, one thing that we can do is think about the distribution of representations in the brain. And even though we focused a little bit on those specific areas of the brain, they all interact. So on top of just this localization of function, we think about the specific functions that are processed by many different areas of the brains of the brain. And this might seem contradictory, but they are complementary in the sense that we experience sensations that are much more complex than often just one area of the brain. And so basically the brain has to co-opt a lot of different areas to be able to think about more complex sensations. And this example that you'll see in the book talks about all the different things that our brains are required to do in order to comprehend a ball rolling across a table. We not only have to look at the shape of the ball to establish that it is in fact a ball, the color of that ball, the motion, so the actual temporal movement of the ball down the table. Then there's depth, so whether or not the ball is moving away, how close the ball is to us, and the location of the ball is also used in order to determine how quickly the ball might be moving. Now, one burgeoning area in cognitive neuroscience has been this focus on neural networks. And we'll talk a lot more about these neural networks in regard to artificial intelligence and trying to represent specific areas of the brain. But one thing that we've accomplished as far as imaging these parts of the brain is through the use of diffusion tensor imaging. And what this does is it not only shows us this dense nerve network and all these neurons that can be activated in the brain, but it also shows us the direction in which the activation is occurring. And what this allows us to do is, rather than just getting a picture of this web of neurons, we're able to establish which neurons are talking to each other. And the importance of being able to attach that information to actual behavior allows us to provide a much broader picture of how the areas of the brain influence the ways that we think. Okay, so that's it for this Chapter 2 video. The quiz has also been posted. I'm going to be trying something new because I had several people who had issues with the Respondus browser. I'm going to be putting a time limit on the quiz. So while you'll be able to take the quiz outside of the Respondus browser, just be mindful of the amount of time you'll have. You'll have 25 minutes total to answer all of the multiple choice questions as well as the essay question. That'll be located on there. And if you have any issues, please feel free to email me, and I will see you next week.